So to be sure, I inform all participants that today webinars will be recorded, and for all the registered people, if it is okay, they have their name in some way will appear in the recording. And all speakers agree to record the presentation. The recorded webinars will be available public. It will be public available on iPlanta webpage. Today, from our number, we were having my last number that I got is 160 registered people. Now we have almost 60 participants connected. I know that some are having some delay, and can maybe wait a few minutes to see if some more people will connect. Generally, we know that people takes a little bit of time before to connect. Rolf, what do you think? We can start. I think that, well, people
Okay,
is, hello? Yeah.
Should I start, Bruno?
No, let me, no, I was just asking.
Okay.
I want to say as a few words so for everybody following the webinars. Please mute your microphone. If not, I will mute your microphone. So please please avoid background rumors. You can keep your camera on, but if the system is too heavy, maybe it's better to have the camera off. Let's say before the end of the webinar, if everybody take the camera on, we can have a virtual group picture. I realize that this is something of interest, and then we can also put everything on the web page. Good afternoon to everybody. This is the first iPlanta webinar. It's the first one for the new year, 2021. I hope that everybody is okay, had a good start for the new year. We still face COVID everywhere, but I hope the situation will start progressing very, very soon for all of us. Today's topic I just introduced with the title. It's specifically related to RNAi suite, a new RNA-based PPV resistant plant. We know very well this all the work done behind RNAi suite. That is the first case of a new resistant, virus-resistant plant cultivar. And during the iPlanta program, it was really, really interesting to know all the progress done and the huge amount of research done on this plant. And it clearly is the first, again, interesting results and probably at a more advanced stage of development of a new potential cultivar for the market. And for me, it is really a great pleasure to have today here Ralph Scorza, that in some ways is the father of Honey Sweet, together with all the other colleagues that have been collaborating with him since many years from Michigan State University. He has been really an experienced horticulturist with long history of working in biotechnology industry, and he has been working in breeding, horticulture, project management, plant patent experience, genetic engineering, and also regulatory system for biotechnology plant, for biotech plant. So, well, again, Rob, it's really a pleasure for me to have connected today. Sorry we cannot meet and shake your hands, but let's do it virtually. And I give you to manage today webinar to tell us at what's the work done and at what stage of development is the Honey Sweet, the PPV resistant plant. Thank you. So you have the floor now.
Okay, thank you. Thank you, Bruno. And thank you everyone for being here. So I'm very excited to be able to participate in this webinar. Since its identification in the early 20th century, plum pox has spread throughout Europe. And even with strict quarantine measures, it has spread to much of the world. It's considered to be one of the most serious diseases of stone fruits, and especially for plum. Plum pox virus is aphid transmitted, and there is no effective control of this disease. And chemical control may be practiced, but it has little value since the virus is readily transmitted simply by aphid probing. So the aphid doesn't even need to feed. Breeding for plum pox virus resistance has been undertaken for many decades, but few, if any, truly stable and high-level resistant fruit varieties have been produced. The cost of this disease is very high, and it is devastating, especially to small growers, plum growers in Europe and other stone fruits. It has been over 30 years since the development of honey sweet plum. At that time, the mechanism of RNAi was not even yet recognized. And certainly, we were very surprised to find that the plant that did not express plum pox virus coprotein was the resistant plant. It was only later that the RNAi mechanism was described in herbaceous models. Since that time, it has been a long road of detailed, meticulous work by a group of dedicated scientists in the US, Europe, including Poland, Spain, Romania, Czech Republic, and also in Chile, all working in the public sector with the goal of bringing good science to protect fruit production for growers and to provide abundant, safe products to consumers. Certainly in Europe, plum pox virus is well known, but the initiation of the development of honey sweet in the US was in the US because it was clear that stone fruit cultivation in the US was at risk from plum pox virus. Indeed, in 1999, plum pox virus was identified in Pennsylvania, and after a 10-year effort, it was declared eradicated, along with most of the stone fruit production in the state of Pennsylvania and tree losses due to eradication programs in neighboring states. Nevertheless, this event taught us that the excellent research that had been done with honey sweet was not enough, because research alone would not put plum pox virus resistant trees into growers' orchards. We needed US regulatory approval. That process was started in 2004. And Dr. Ann Callahan at the USDA put much of the molecular data together for the dossier, which was finally approved, honey sweet for cultivation in 2011. So that was 7 years later. Since plum pox virus has not reappeared in the United States, there is not a great demand for honey sweet commercially, but it is being used as a germplasm resource should plum pox virus ever again enter into the US. But what about Europe, where plum pox virus is a devastating and spreading disease of plum and other stone fruits? Well, honey sweet has in fact been field tested in Europe since 1996, It's almost 25 years of field testing in Europe, and has shown extremely high and stable levels of plum pox virus resistance under heavy natural and artificial inoculation pressures. The barrier to cultivating this uniquely valuable plum variety, and also for using it as a germplasm resource for breeding additional plum pox virus varieties in the plum pox virus resistant varieties in the EU, depends on the EU approval process. So this is the critical next step for the European Union, not for honey sweet alone, but as an example for all other horticultural crops that are genetically improved through RNAi and other new genetic technologies. Although frankly, RNAi is well tested and certainly not a new technology anymore. Today's speakers will focus on the RNAi mechanism in honey sweet, long-term field trial results in the EU, health and environmental safety studies, and the latest update on the honey sweet dossier being prepared for submission to the EU regulatory authorities, and PRESCA, the association that will shepherd the dossier through this process. So I join in thanking you for attending this meeting. As Bruno said, please set your microphones to mute. And if you can conserve bandwidth, you might want to turn off your video. Also, please submit any questions or comments, brief ones if possible, through the chat box, and we'll select some of those questions after each speaker's presentation, and then save some also for the end of all the talks. So with that, I would first like to introduce Dr. Michelle Revolundro. Dr. Michelle Revolundro was, until his retirement in 2019, Director of Research at INRA Bordeaux. Michelle's research focused on the control of plum pox virus and plum pox virus control, and he is presently a French core group member of iPlanta. Michelle is one of the earliest researchers to work with honey sweet plum, having begun his work with honey sweet in 1990, studying and identifying the siRNA resistance mechanism, again, even before it had been identified in herbaceous model plants. He has coordinated honey sweet projects in Europe since 1996. Thank you, Michelle. And Michelle, I will indicate when you have about 2 minutes left to speak. So it's all yours.
Thank you. Thank you very much, Ralph. Okay. Did you see my screen or not?
Not yet.
Screen
Not yet.
Now it's okay?
Now it's coming.
Good.
Okay. Put the full screen on your computer.
Okay. Thank you very much, Ralph, for this nice introduction. And the other point too, I would thank Bruno about this special webinar with Honestly. It's really interesting to have this model, this example. On the title of this presentation, you have, let's say, two keywords. You have the Sharpa disease and RNAi. I just focused on that 2 points. So the next slide is showing the occurrence of the disease in the world. Of course, you have the green paint showing that all of those countries are conserved. You can nicely find out that Oceania is the southerly continent non-affected by PPV, of course, Australia and New Zealand. And I just, attract your interest on this red spot. The red spot is Bulgaria. Bulgaria was cited as the first country to be the source of plum pox. And if the disease was expanded like we have on this world map, then we should consider two things: the long distance spread of the disease and the short distance spread. What means the long distance? The long distance means that the infected material have been transferred in those areas, and the worst too is the latency of the disease. You can have what they say, imported the material, you can have set it in your orchard, but the disease only shall occur after 10 or 20 years. That's long, and that's really critical when we are speaking about the PPV spread in the world. However, about the short distance, this is the effect that Raf titled. About PPV, about the Plum Pox virus, it is transmitted through the non-persistent matter. It means that it's very passive, and we can understand why the disease, the virus can cross borders and then reach to any parts of the orchard. This is just a scenario. Then when the aphids, I say, suck on the plant cells, then the aphids doesn't know, of course, if it is veriferous, but it transferred the virus that is spreading in our trees. That's the consequence. The consequence is the impact on fruits. You can have deformed fruits. You can have those typical marks on the apricot stones or leaf deformations, or here also gummy plum or some fruit dropping. When we have that, then we need to control. And the European Union spend lots of money to track the disease. This is quite of a similar situation with the COVID-19 that we have on today, suggesting that we need to test, we need to track the virus. And after that, then you need to quarantine the area. For us on human with the COVID, we have the lockdown, but the quarantine zone means the lockdown. But of course, we cannot kill human, but we can kill trees. And we remove trees. We also develop, virus-free materials. Otherwise, we cannot control the disease. Does require some cost. Does require some money. And with the charcoal project, we develop all of those tools. And the other aspect too, when we compare with the COVID-19, how humans have the antibody production, but plants doesn't have. So what we have to find out? So you can have some natural source of resistance, and that's lead to the breeding technology. So exploiting some natural gene resistance. Let's compare just for crops, for commercial crops, highly exploited as well in Europe or everywhere in the world. Here you can have that all commercial cultivars are susceptible, except with Prunus avium. With cherry tree, it's so strange. We have only the rootstock, cold or pontavium, that can show symptoms. But otherwise, the commercial cultivar recovered. Yeah, it's very strange, but that's the situation. And when we are looking those commercial cultivar, including apricots, plum, and peach, then you can have those natural genes involved in the plant metabolism that interfere with the virus replication. Among the typical one is the yo-yo. The yo-yo is showing the hypersensitive reaction. So some comparative work have been done with only sweet, but it's different. what they say, the genes involved are encoding plant proteins and interacting too with virus protein. It's indirect interactions with the virus. This slide summarizes the situation. We have the Spanish colleagues, Mariano Cambra, took calculators with these colleagues and then demonstrated that shaka is causing really yield loss. And in this paper that they published in 2006, they say that we have 10,000 million euro of deficit caused by shaka disease. That's really amazing. And what's the links with iPlanta? With iPlanta, if you follow some talk with the preceding webinar, then you watch the role of the viral RNAi. The viral RNAi can interact and can interact with the virus, but that's when you are using the topping technology when you spray. And when you use that technology with Plum, that the viral RNA is inside, it's impossible. That's the reason why we engineer the RNAi technology in the RNA-Suite Plum. And the nicest with RNA-Suite Plum is developing a stable, a durable resistance that can be transferred, easily transferred to the hybrids, to the progenies. And that's the nicest with this technology, RNAi.
2 more minutes.
Finish soon. Just for reference, when you have here the comparison between susceptible leaves and resistant leaves, you have with the conventional crops, the viral RNA that is replicating in cytoplasm and giving the viral particles. And that leads to the transformation of the metabolites. To end, that's the key point. The key point is the double-strand RNA that is what they say engineered, slice in the cytoplasm, and then targeting the viral RNA. The viral RNA is degraded, and this is the main device. The RNA-dependent RNA polymerase 6. This is amplifying the small RNA and leading all time to the reduction of the viral RNA. The RNAi is a viral ship. This is a successful achievement between French and American. And now let us to hear Dr. Juan Zagre telling more about the effect of RNA-Suite in European Union field. Thank you very much for hearing me.
Thank you, Michelle. That was excellent. We appreciate that. The next talk will be by Dr. Juan Zagre. Dr. Zagre graduated in agronomy by the University of Agricultural Sciences, Veterinary Medicine in Cluj-Napoca in 1994. He obtained his PhD degree in 2006, in 2003 in plant breeding, investigating plum resistance to Plum Pox virus and its breeding by conventional methods. Since 2004, he has become the senior scientist, and during 2004 to 2019, he was head of research department and member of the board of directors of fruit research and development in Bistrița, Romania. Starting from December 2019, he became the director of FRDS in Bistrița. His field of expertise are plant virology, fruit breeding, plant certification, and GMO field trials. During the last 2 decades, along the last 2 decades, he conducted different research projects in these areas of study. Since 2012, he's been member of Romanian Biosafety Commission. Since 2017, he's become associate member of the Romanian Academy of Agricultural and Forest Sciences. Dr. Zagre is the author of over 90 publications and international reviews or peer-reviewed conference proceedings author or co-author of 10 books or chapters in books, and co-developer of 8 plum and 2 sweet cherry varieties in Romania. So with that, Ioan, we'll hear your talk. Thank you.
Thank you, Dr. Scorza, for introducing me. First of all, I like to thanks to the organizers that accepted my talk within these webinars. My presentation entitled 2 Decades of Honey Sweet Field Trials is actually an overview on long-term field trials of the first RNAi genetically modified plum-resistant Plum Pox virus. The field investigations were undertaken in different European countries and also in USA. As was already presented, Plum Pox virus, which causes Charcot disease, is the most detrimental viral pathogen of stone fruits because it often produces severe symptoms on fruits and premature dropping, making thus plum fruit production very problematic. Therefore, the active research to control Charcot disease remain a major interest for breeders for obtaining PPV or resistant plum as the most efficient solution against PPV. As was mentioned by the previous speakers, the pathogen-derived resistance strategy used as a complementary approach at conventional breeding permitted to develop the notorious C5 subsequently named honey sweet, which is an RNAi-based This plum is the results of a mutual cooperation between USDA and INRA Bordeaux, France. Once its highly resistance was to graft and aphid-mediated inoculation was shown under greenhouse conditions, the experiments were moved in the field in different endemic areas from Poland, Romania, Spain, and Czech Republic. To perform the trials, an international honey sweet working group, including public institutions and scientists, mainly from US and European Union, was set up. The field investigations performed during more than 2 decades consisted of 3 pillars. The first pillar of investigation was related to the resistance, precisely to evaluate the resistance of transgenic plums in the field conditions under high pressure of infection with PPV, and to assess the stability of PPV resistance in honey sweet under the coexistence of PPV and 2 heterologous ILR viruses. Because the potential risk have been expressed with the field release of virus-resistant transgenic plum, a second pillar of investigations was related to some concerns such as potential risk of transgenic plums containing CP gene on the diversity of PPV populations, the potential impact of honey sweet on indigenous aphids populations, the potential bees preferences for transgenic and the conventional plants, and the potential gene flow consequence, precisely the gene flow from transgenic to conventional plants, affecting thus the coexistence. Once the objectives of the 2 first pillars were achieved, The experiments were extended to other pillars in order to gain information on the agronomic performance and to assess the possibility to reduce the number of insecticide treatments against aphids. To evaluate the resistance of versus conventional plums was assessed. The monitoring of disease spread based on visual observation and virus infection was confirmed by serological and molecular tools. For example, field trials in Romania revealed that the temporal spread of PPV recorded a continual increase in infection in conventional plum in all the 3 experimental plots, getting to none of the transgenic plum honey sweet was infected. Important to mention here that similar results were obtained in Spain, Poland, and Czech Republic. Taken together, this data, honey sweet clearly demonstrated its high and durable resistance to natural PPV infection in endemic areas. To assess the stability of PPV resistance in honey sweet upon PPV infection and co-infecting with heterologous viruses, trees of honey sweet were body inoculated with PPV alone and with a combination PPV plus PDV and PNRSV. Regardless to singular or mixed infections, PPV could be observed and detected only near of inoculum sites. This the plants. Conversely, obvious PPV symptoms could be observed on the shoots developed from the inoculum buds, which secure a permanent and high infection pressure on honey sweet. Overall, when artificially inoculated with PPV and in combination with heterologous viruses, systemically spread in honey sweet trees, while PPV remain close to the inoculum site, showing thus an effective inhibition of virus replication. Here also, it should be mentioned that similar results were obtained in 3 countries, Czech Republic, Spain, and Romania. Based on the results obtained on the first pillar, we concluded that regardless of the different conditions of the field testings, honey sweet could not be infected by natural aphid transmission, exhibiting a high and durable resistance to PPV. When artificially infected, PPV could produce only a poor infection in honey sweet, independently of the presence of the challenger virus, but the infection did not prosper due to the silencing mechanism. And hence, there is no evidence that heterologous viruses could affect the stability of the engineered protection in transgenic honey sweet plums. Because the potential risks have been expressed with the field release of virus-resistant transgenic plum, a second pillar of CPG, it was expressed the potential hazard concerning the emerging of transgenic plums containing CPG on the diversity of PPV populations by comparing the variability of PPV isolates detected in transgenic plums versus isolates selected from conventional plums and surrounding the transgenic plums. Sampling was initially based on PPV symptoms on leaves, and virus infection was confirmed by serological and molecular tools. PPV strain determination was serological achieved by TAS ELISA using monoclonal antibody, while the molecular strain typing was done by ICRT-PCR targeting different genomic sections combined with RFP analysis. The characterization of the isolates was completed by sequencing of PCR products. Here we have the example of investigation in Romania, where the results show that the ratio of PPV strains in transgenic and conventional plums surrounding the transgenic plums was approximately in transgenic and PPV or nucleotide sequencing show that these isolates were very similar in transgenic and conventional plums. Similar results were also obtained under Mediterranean conditions by our colleagues in Spain. To assess the potential impact on the diversity and dynamics of indigenous aphids populations, we monitored the winged aphids that visited the two types of plums, transgenic and conventional plums, by using sticky shoot methods. Then a comparison of number and species of aphids landing on transgenic and versus conventional was made. The results in Romania, for example, show that both the flight curve of aphids and the percent grown in the same experimental plot was approximately similar. The similarity was also found in the number of aphid species and Mediterranean conditions in Spain. The assessment of the potential bee preferences for transgenic or conventional plants was made along 3 consecutive years during blossom times, precisely when it overlapped on the 3 varieties, 2 conventionals and honey sweet respectively as transgenic. Bees visiting transgenic and conventional plants were monitoring by each of the day, each 5 minutes. Although there were some fluctuation on the number of bees that visited the 3 varieties year by year, the overall results show that there were not significant differences between the number of bees that visited the transgenic and conventional plants. Hence, bees do not make any difference between transgenic and conventional plants. GM flowering plum has some unique qualities when compared with other field crops, such as soybean and maize. GM flower from pollen, when it occurs, affects the seeds, not the fruits. Fruit receiving GM pollen is not changed because the pollen enters the embryo of the seeds. Plum seeds are not used for foods, and the seeds are usually destroyed since plum are usually propagated, reproduced by grafting. In cultivation, in orchard, when seedlings appeared, they are killed by herbicides and cultivation. Just in case, if a growers use the seeds to obtain the rootstock for grafting, these rootstock usually do not produce fruits. In addition, an experiment performed in the United States showed that spatial and temporal pollen and seed-mediated GM flower from GE plums support the feasibility of coexistence of GE and non-GE plum orchard, because the pollen can be obtained within the second pillar. We concluded that serological and molecular variability of PPV populations in transgenic and conventional plants provided evidence that the transgenic plants expressing PPV-CP gene do not promote recombination and therefore do not represent an environmental risk for new emerging PPV strains. Honey sweet transgenic plum has a neutral effect on the diversity and dynamics of indigenous aphis populations, and there is a substantial and non-GE plum orchard is feasible. While the field trials within the first two pillars clearly demonstrated its resistance to PPV infection and safe for the environment, expanded the studies on agronomic performance of honey sweet plum were requested. Thus, a new experimental plot including transgenic honey sweet and two largely grown conventional plum varieties, Stanley and Renclo-Dalton, was studied for data collection. Gaining information on the agronomic performance of honey sweet was related to the yield potential and the quality of fruits, biometric attractiveness, and nutritional value, respectively. The average yield on three consecutive years revealed that honey sweet produce a yield like a Stanley-like yield, while Renclo-Dalton had a significant lower yield. Since Stanley is already well known as a very productive cultivar, cultivars. The average fruit weight and size highlighted the superiority of honey sweet when compared with the two conventional plum cultivars grow in the same plot. Expectedly, the stone weight was the biggest in honey sweet, but interestingly, the percentage of the stone in the weight of the fruit was very low, similarly with that in Renclo-Dalton, which is with much lower values than on Stanley. In terms of attractiveness, honey sweet fruits have a positive impact because the size of the fruits is clearly superior to the other two varieties. The stone is small, related to the weight of the fruits. The shape is oval, quite regular. The background color of the skin is deep purple, uniformly distributed, and the pulp yellowish green color is firm, very appreciated. The nutritional value was assisted by determining the dry matter, carbohydrates, protein, lipids, mineral, vitamins, and polyphenols was determined in honey sweet and in at least one of the two conventional plum varieties, while the content in lipids and the protein was higher in honey sweet than in the two conventional plums. The content in minerals was also generally higher in honey sweet than in the two conventional cultivars, while the content in the amount of vitamins determined in honey sweet was similar in at least one of the two conventional cultivars. safety issue related to the use of pesticides and will need to comply with EU requirements to reduce its use for crop protection. In this context, we have raised the questions as to whether some treatments against aphids vectors might be avoided in plantings of honey sweet, since it cannot be naturally infected by the virus mediated by these vectors, and consequently its presence in the orchard do not into account that avoiding some treatments raises also additional questions of controlling other important insect pests in the same time with Lasperesia funebrana. Wind aphids were monitored by using sticky shoot methods, and in parallel, adults of Lasperesia funebrana were captured by using specific pheromone traps. The concurrent analysis of the flight curve and the presence of the pests revealed that the flight curve of Lasperesia funebrana overlap most part of those of aphids. Consequently, treatments with insecticides are necessary to control Lasperesia regardless of its needs against aphids. However, Lasperesia funebrana flights ended in September, while aphids flight resumed and can spread PPV in susceptible and tolerant cultivars, but not in honey sweet plantings. And hence, there is the possibility to avoid treatments in this period. Based on results obtained within the third pillars of the field trials, we found that when into the high yield group of cultivars. Similarly, the quality and the nutrition against aphids are not necessary in honey sweet plantings in September, when aphids are resuming their flight for a short period, and hence a slight decrease in the number of treatments with insecticides could be possible without promoting PPV spread. Avoiding the application of insecticides against aphids in PPV-resistant transgenic plant plantings could also be possible if the populations of other plant pests, excepting the aphids, of course, controlling the same time are under the economic threshold of damage over when other eco-ways, for example, pheromone traps for controlling these pests are available and applicable, conducting thus to a significant reduction of pesticide use. We also have a few general conclusions based on the long-term field trials. Quality and productivity. Honey sweet is not only protected against PPV, but also has the attributes of fruit quality and yield that make it suitable for commercial production. Field trials performed in different countries clearly demonstrated the neutral effect of transgenic plants carrying the PPV-CP gene on the diversity of PPV variants and non-target organism. I talk here about the aphids and bees, thus making it safe for the environment. Potential for genetically engineered perennial crops, as like for plum, to be an integral part of IPM programs to reduce environmental and health impact of pesticide use. Of course, taking into account this large amount of data and results, presently the real challenge to practical application of RNAi mechanism of honey sweet remains approval in European countries highly affected by PPV. Thank you very much for your attention.
Thank you, Yuan. That was really an excellent, very wide-ranging report. Whereas one question that came up on the chat, is it possible to have resistance using only a transgenic stock and putting on a non-transgenic scion so that the plum fruit would be non-transgenic? Can you address that?
Could you repeat the question, the second part of the question?
Okay.
Resistance?
Okay, so the question is, could you use honey sweet, for example, as a rootstock that would give plum pox virus resistant to a non-transgenic scion grafted onto it?
Okay, I understand. We didn't test this kind of aspects, but if I well know, there is an experiment in progress in Italy, which tests this kind of possibility. I have no idea about this information.
Well,
I cannot provide this kind of information this time. If the transfer of the resistance is possible from the rootstock to the variety, to the site.
Okay, well, I can answer some of that. It appears from work that we have done in the USDA that it does not appear that functions to give that honey sweet would give resistance to the scion that was grafted onto it. I don't know if Chris, you have, or Ann Callahan, if you have anything to add to that.
But honey sweet.
Okay, well, one more, since we have a little bit of time, we'll have one more question here. There's a question concerning honey sweet and conventional plums. Even if the genetic material of the honey sweet pollen would only be found in the seeds of the fruits emerging from outcrossing, in my opinion, these fruits could not be sold as non-GM. So if a fruit that's from a non-GM tree has a seed that is GM, could that fruit be sold as non-GM? I don't know the answer to that. Does anyone have any?
A potential crossing between honey sweet and the conventional plum cultivars do not make fruits of conventional cultivars to be GM. Of course, the contamination between brackets can occur only at embryo level, so the flesh is not affected. In this way, there is no risk to grow transgenic and conventional plums very close.
Coexisting, you mean?
Right, coexistence.
Yes.
But I guess it would be up,
is really visible.
I guess it would be up to the regulatory agencies to say if that conventional plum with a transgene in the seed would be considered transgenic fruit. I guess that would be a regulatory question, and I don't know if we have anyone here who could answer that.
You actually have a 0.9 level under which your product does not need to be labeled. So if 0.9% of a product is GM, you don't need to label.
Okay.
But of course, this only can be applied for authorized products. So if you actually would have contamination from a non-authorized event, then everything would have to be destroyed.
Okay, great. Well, thank you.
Hey, Ralph.
Yes.
From your pollen flow study, though only in 4 out of 11 years did you ever see the pollen move further than one tree. Is that correct?
Yeah, further than I forget how many meters, but it was the pollen flow is not very very great in plums. So yeah, I think if certainly it would, if two orchards could be very easily separated, one orchard being honey sweet, another orchard being non-transgenic plums. I don't remember right offhand how many meters distance they would have to be, but it's not very far. So the plum pox, I mean, the plum pollen does not travel very far. It's not wind, it's not moved by wind, it's moved by bees, and bees have a very specific way that they travel in an orchard. So we found that there was very, very little movement of transgenic pollen out of the orchard, out of the honey sweet orchard. And we have another question. Let's see if we can get this one too. Okay, I think I answered that. The question was: is plum pollen only carried by wind or also by bees? It's not carried by wind to any significant amount at all. I would say it's not carried by wind at all, but carried by bees and other pollinating insects. So with that, I think anything else anyone wants to say about that?
May I answer, Ralph, to the first question?
Sure.
We did this kind of work, and unfortunately, we cannot have, siRNA movement or diffusion in the non-transgenic shoot. Unfortunately.
Okay. Okay. Thank you very much. Appreciate that, Michelle. I guess our next speaker will be Dr. Chris Dardik. Dr. Chris Dardik is lead scientist in plant molecular biology at the USDA ARS Appalachian Fruit Research Station in Kearneysville, West Virginia, where he has worked for the past 15 years. He received his PhD from the University of Maryland in plant virology in 1999. He currently serves as lead scientist for the Genetic Improvement of Fruit Crops Unit, whose mission is to provide genetic solutions to major problems affecting temperate tree fruit agriculture, including orchard productivity, high labor costs, bloom time related cold injury, consumer demand for superior fruit quality, as well as the need for new genetic engineering technologies to address emerging threats. So with that, I'll hand it over to Chris.
Thank you, Ralph. Hold on one second. I'm trying to get my screen sharing up here. Oop. Sorry about this. Can you see my screen?
Yes.
Okay, there we go. All right, so thank you very much for the introduction, Ralph. I'm going to talk a little bit about some of the more recent data we've had on the molecular characterization of honey sweet, along with some health and animal studies that were done as well. So let me just start with a little bit of background information on honey sweet. We had known previously from work that Ralph had done along with others on the that the insertion event in honey sweet was a bit complicated. So at the top of this diagram, you can see sort of the vector that was used between the agro borders. You'll see an NPT2 cassette, the PPV coprotein cassette, GUS, and then some additional vector sequence. So when that actually went into the plant, it turns out there was actually two insertion events that were nearby. So they're on the same chromosome, some distance from each other. One of them is a rearrangement of many different pieces. And then the other turned out to be serendipitously a hairpin arrangement. So you can see here that we have two copies of a coprotein in a sort of tail-to-tail arrangement, which we presume is responsible for the high levels of resistance that we see, though I don't think we've proven this definitively just yet. So one of the challenges of working or getting molecular data in this situation is that it is complex. So we needed to get a lot of information both about these inserts and their potential impacts on the genome. So one of the things we had done in response to that was we had actually sequenced the genome of Prunus domestica, which is a hexaploid. Now the genome we sequenced was actually from French, which is a most commonly used cultivar for prune production throughout the world. And we produced what we call a phase genome, meaning that we had all the individual chromosomes kind of delineated to some extent on separate contigs. So if we search those for the regions of the insert, so we had some short flanking sequence from Illumina short read data, we could position these roughly on these different contigs. And these are potential positions. We couldn't determine exactly which subgenome or which copy the insertion events are within. And so that's something we're still trying to clarify. We've recently used nanopore sequencing to get long molecule reads for HoneySweet. And we are now in the process of analyzing that data. So we should have that information before long in terms of exactly which chromosome position these insertions are and how far apart they are from each other. So from the data we had, one of the things we were able to do, and this was work done by Ann Callahan, was to look at the potential impacts of the HoneySweet insertion events on flanking genes. So in this diagram, this center line, if you think of the x-axis here as the position along a chromosome, the blue line represents the insertion point for insertion 1. And then on either side, we have the plum DNA flanking it. And so this is RNA-seq data where the reads are aligned to the genome. And you can see the positions of the flanking genes. So one of the things that we were able to show is that if you look at the top 3 bars here, these are from fruit and the bottom are from leaves. And it turns out there's really no impact on the expression levels of the flanking genes, and we don't see any new ORFs or odd expression patterns flanking the insertion. So really the insertion really doesn't seem to have a significant impact on the flanking DNA. One of the things we've been really interested in assessing is the small RNA component in HoneySweet. So in one of the unique things of RNAi technology is that you're not producing protein, but rather small RNA is the active ingredient. So there's been a lot of discussion and in some cases controversy over the last decade or so about whether small RNA could be potentially harmful or not to either the environment or consumers. So we've spent a lot of time looking at small RNA expression. And what I'm going to talk about here is 2 data sets. So one of these was a multi-location trial, if you will, where we collected HoneySweet and a comparator, Stanley, a non-transgenic comparator that's susceptible to Plum Pox virus. And we looked at the siRNA profiles both in PPV infected fruit, healthy fruit, and in HoneySweet. And so the trees that we used came from these 4 locations. And the one thing I just will note here is that in the US, since we do not have PPV, we know that these have not been even exposed to PPV. So this was just one of the controls and comparisons we tried to include. The second location comes from a planting in the Czech Republic. So this is a really great field trial that had been put in that has HoneySweet trees that were grafted with infected limbs from a non-transgenic cultivar, Emma And in some cases, those limbs were allowed to stay on for a number of years and then were removed. And in other cases, they were just left on. So we were able to collect siRNA data across all of these trees. Okay, so in that trial, just to give you some idea of what was observed, the HoneySweet leaves, if you look at over time, just sampling for Plum Pox virus infection, the trees remain virtually PPV free. So there's very little or very low levels of Plum Pox virus ever detected using qPCR. And this is just for comparison. This is the infected Emma-Lapperman grafts. So this just kind of shows that honey sweet resistance is extremely durable, even when you have fully infected large limbs grafted to it, that it never seems to succumb to the virus infection. The other thing I think this somewhat addresses was the question about grafting. And conversely, we never saw, this data shows that there was never any real resistance conferred to the infected limb. So it was unable to cure at least an infected limb. So looking at the small RNA from a global perspective, and this was done through small RNA sequencing approaches. So we sampled them, extracted small RNA, and did bulk sequencing. And just looking at the distribution, size distribution of small RNAs, what we found was that in honey sweet, and this was somewhat known, it pretty much confirmed prior data from blots and so forth, that honey sweet uniquely contains a much higher level of 24 base pair small RNAs. And these are presumably associated with the resistance. In infected Stanley, so this is plums infected with Plum Pox virus, we found that the virus results in a high level of 22 base pair small RNAs. So you don't really see the 24s when they're infected, but these 22 mers. So this is the major difference in the profile between honey sweet and infected plums. And one thing we've been able to do is sort of based on the read counts, we can kind of quantify how much small RNA that's derived from the coat protein in honey sweet relative to what you might see normally in an infected tree. So in the first column here, you see Stanley. Obviously, these are uninfected trees. There's no reads against the Plum Pox virus itself or the PPV coat protein. And this is all from fruit. If we look at infected Stanley fruit, if we look against the entire PPV virus, 35% of all the small RNA that we found in the fruit was from the virus itself. In just directed against the coat protein was about 2%. In honey sweet, we see this fairly consistently, that about 0.3% or so of the small RNA was due to the transgene matching coat protein. So what this is telling us is that in an infected tree versus a honey sweet tree, there's about 6 or 7 times more small RNA derived from coat protein from virus infection than there is from the transgene. And if you consider the entire virus, you're getting a consumer would be getting a substantial amount of small RNA from that fruit that's just simply due to virus infection. So this was important to put into kind of context the small RNA produced from honey sweet and just how much the consumer would be exposed to relative to the alternative, which would be consuming potentially infected fruit. These are just some numbers from both sites showing the actual read counts per million for small RNAs. So on the left, we have the Czech Republic plot, the graft inoculated trees, and on the right, we have the multi-location trees. And you can see that from honey sweet, and this is all leaf data, we do see a substantial number of reads to the PPV coat protein region, which is not surprised. We expected that at some level. We also see some reads to GUS. So there's a small number of reads matching to that reporter gene that's in the cassette. Not really anything to NPT2, maybe a small number of reads to this flanking vector sequence. And in relationship to that, you can see from like the example here, the inoculum, just how many reads are span the viral open reading frames themselves. So this just gives us some context again for the small RNA expression levels relative to what is produced in the tree naturally. The other thing we were interested in was whether the small RNA expression was uniform across the transgene or whether there were perhaps hotspots of certain regions producing higher levels of small RNAs than others. Again, this was to give context for, are there certain specific sequences that may be very, very abundant that we should be looking at. So on the top of this figure, you'll see a hairpin diagram of the coprotein, like I said, tail-to-tail insertion event. And in red is highlighted the coprotein open reading frame. And on the graphs below, top one is honey sweet from the EU. Each of these dots represents a specific small RNA species or sequence. And the dots, whether they're derived from the positive or negative strand, are increasing in expression value. So you can see in both directions. So these sort of regions where we see a number of small RNA species that are much higher levels than the others. And we do see these kind of hot spots. I have kind of arrows down at the bottom. So whether it's from the EU, the US, or even naturally infected, PPV infected trees, these hot spots seem to be somewhat uniform. So there isn't any, doesn't appear to be any significant change in that distribution. One of the other things we were able to do with this data was to look at in these infected limbs that were grafted onto honey sweet, and they had been there for a decade, to look at the viral populations to see if there's been any selection pressure from imposed by honey sweet. So potentially due to the movement of small RNAs into the infected limb or possibly other mechanisms. So from the small RNA data, we were able to reconstitute actually the genomes or the populations within these infected grafts and look for SNPs. And so in green here, you see the Plum Pox virus genome. And then below that, you see the SNPs in each of the grafted trees. And if we look at the coprotein sequence, we don't really see any evidence of accumulation of SNPs indicative of the potential for the virus to be adapting to the resistance imposed by honey sweet. So, so that implies that at least from that kind of interaction, honey sweet doesn't seem to be imposing any kind of strong selection pressure on viral populations. Now I'm going to switch gears a little bit and talk about the animal feeding study we did. So this was done at Oklahoma State University with mice. They had this, Brenda Smith had a lot of experience working with plums and feeding mice plums, which it was very fortunate that they were able to do this. So the study involved 150 4-week-old mice. They were split 75 male and female. We lyophilized all the fruit and it was mixed with diet. So it was mixed at 2 concentrations. So 5% of the diet being honey sweet and 15% of the diet being honey sweet. We didn't go higher than that because a honey sweet has some, or plums have laxative properties and things like that. So if you get up to 25%, you can start getting health effects just due to plum. So we wanted to stay in the lower range. And this is also significantly higher than any consumer might be experiencing in terms of the quantity of plums we eat. So the mice, we had a no plum and 2 different concentrations of honey sweet and Stanley plum. And then the food intake was recorded and body weight. And this was a 90-day study followed by toxicological and pathological analyses. So I'm going to skip through this a little bit rather than going through all the details. This is just showing body weight. There was really no change in the body weight of male or female mice, regardless of whether they had honey sweet, plum, or Stanley. And this is some of the kind of chemistry data that was obtained. This is, white blood cell differentials. One of the things I'll point out here is the p-values. In almost all cases, the p-values were insignificant. There were a few instances where we had significant p-values, but the values in terms of the chemistry were within the normal range you would expect for these animals. This is some additional blood chemistry. This is just showing here for what was done for the females. I am not a medical person, so a lot of this stuff I'm not completely clear on. But again, just to point out that really we saw nothing significant at any dose of plum relative to the controls. It just didn't seem to have any significant impact. The other thing that was done was a pathological analysis. So the animals at the end were dissected, and virtually every aspect within the animal was looked at. This was done by a team of people that looked at and recorded each one for whether or not it was potentially abnormal. So abnormal being something that looks outside the range of what they typically would see. And you can see here that this again is just for females. There was no difference in the pathologies for across the scale in terms of anything associated, particularly with honey sweet plum. One of the other things we were able to do was sort of combine this data. So since we had sequenced the small RNAs, we knew kind of what the levels were within the fruit that were fed to the mice, and we could pick out potential off-target effects that could be predicted. So we blast searched these small RNA sequences against the mouse genome and looked for potential matches. So I think we had a minimum match of 18 and a certain number of mismatches. I don't remember exactly what the parameters were used for that. I apologize, but it was really a very low stringent analysis just to say what could we hit. And there were really 3 genes that popped up that could potentially be off the effects of, or could be off-target effects. So one is this MYOF gene. Mutations in this result in muscle defects in muscle regeneration and angiogenesis. This NT5E mutations result in calcification of joints and arteries and alterations in dendritic cell infiltration. And then the last one, LRP6, has been associated with Alzheimer's, cancer, and osteoporosis when it's knocked out or downregulated. So we looked within the mouse data. They went back and looked for evidence of any of these kinds of pathologies associated with the mice, and they didn't really find anything. So within this study, there was no statistical or anything significant in terms of differences between consumption, and there was no evidence of really off-target effects that could predictably lead to any kind of problems or pathologies. So just to summarize, honey sweet has a complex multi-site insertion event that has really no measurable changes in the flanking regions. And again, we're continuing to characterize that. Part of the impetus for that has been a recent submission to Canada for import approval of honey sweet. And as part of that, we're looking to get some more complete information for the honey sweet insertion. The siRNA expression in honey sweet is relatively stable under different environments and over time. So we do not see any dramatic swings in expression. We had some recent data from a separate project showing that actually the small RNA or the RNAi machinery seems to be induced by Plum Pox virus in the phloem of leaves. And that happens during the spring when the plants are leafing out and the virus is reinfecting at about 6 weeks post bud break. But in our data, we had leaves collected in some cases at different times, and we just didn't really see anything dramatically different in regard to siRNA expression. Viral infection level of plums produces significantly more siRNA than honey sweet. So this is obviously important for regulatory consideration in that people have been consuming Plum Pox virus RNA for some time. So what you're being exposed to in honey sweet is not significantly more than that. Again, this study in the Czech Republic showed that PPV populations in limbs grafted to honey sweet really don't show any adaptive evolution. And this mouse feeding study, we didn't really see any measurable health effects on honey sweet plum consumption. So just acknowledgements. A lot of people have been involved in this. I just want to point out a few. Our team here at Kearneysville, and Ralph, and a postdoc, Kelsey Galimba. And at the Czech Republic, Jiban, Kushwat Singh, and Yaroslav Polak did a tremendous amount of work on their plot in terms of helping us get the molecular data and analyzing it. Brenda Smith at Oklahoma State University, Tadeusz Malinowski in Poland, who helped with the early small RNA analysis, and then a number of labs here in the US that helped with some of the genomics and bioinformatics, as well as Michelle and Mariana, who had been working on these on HoneySuite for a long time. So with that, I will stop.
Thank you very much, Chris. It was a really great talk. Appreciate that very much. There are a couple of questions, but I think perhaps we'll save those for the discussion after the next couple of talks. So we'll just move on, and I hope that's okay with everyone. But I think we'll have time at the end to answer or discuss all these questions. Okay. All right. So the next, our next speaker is Dr. Nick and Van Guel. I'm not sure how to pronounce your last name, Nick. I'll just call you Dr. Nick right now. Recently obtained his PhD at the VIBU Ghent Center for Plant Systems Biology, where he studied the mechanisms of lateral root development using genome editing with CRISPR. Meanwhile, Nick has gained experience in regulatory aspects related to plant biotechnology, as well as teaching plant biology and science communication and other outreach activities. So,
Ralph, this is Pete. Ralph, this is Pete. Before you try to pronounce Nick's name again, you skipped me in the program.
Excuse me?
You skipped me in the program.
Oh, I skipped you. Oh, okay. Sorry about that.
That's fine. That's fine. Okay. Give me extra time to pronounce Nick's name again.
Oh, well, maybe you can help me out when I get to that point.
I'll be happy to do so. So would you like me to pick up here?
Please do.
Okay, let me start with sharing my screen. There we are. There we go. And this has the advantage for me that I can introduce myself. My name is Piet van der Meer, and as you see here in this little bio I've pasted under my name, I have for most of my life been involved in biosafety regulations, GMO regulations. And I'm one of those people who are just enjoying being part of this work, volunteering time to do this. My input is not as much about the scientific part, but more about the regulatory part. And that is related to something that Ralph said in the beginning, that this HoneySuite dossier can be a very nice test case. The reason of my interest is that I have been involved in these regulations since the first drafts we received on our desks in 1987. Then directives were adopted in 1990, the first GMO directives. And that whole system has changed many times. And most recently, we have seen another change to the whole system. This is a regulation about transparency and risk assessment, which will come into force soon. The situation is that since 2012, I guess it is, there have been no submissions for cultivation, so for placing on the market for cultivation in Europe. So there is, as a regulator, it's always interesting to see how well regulations work. There has been no test. So this case is a very interesting case, and especially because it has a number of very new things in it. First of all, this is the first product submitted for placing on the market in Europe, developed by the public research sector. The general assumption is that only very big multinationals can go through this system because the bar is so high. So this will be an interesting test to see how this works. Secondly, this is the first time, or one of the very few first times, that instead of annual crop plants, which are the majority of the dossiers that have been submitted, this is a perennial tree. This is a RNAi, which is quite new. And this is a dossier for both cultivation and consumption. And then the particular part here is that the consumption will be consumption of an unprocessed product. For me, the reason to participate in this dossier is the part that I like the people, is that I'm quite keen to see how this dossier goes through the system, how it works, and to share the experience with this whole process with other public research sector institutes. So we will make an online system whereby we share real-time questions received from authorities or from EFSA and responses given. Then a brief comment about the dossier submission, because there are several ways you can submit a dossier to the system for placing on the market. If it is a product just for food feed, for example, for import, then you submit a dossier under Regulation 1829. Is it only for cultivation, such as ornamental flowers, then it's the Directive 2001/18. And if it's a situation of both food feed and cultivation, then there are two options. Either you submit the whole dossier, both food feed and cultivation, under Regulation 1829, 2003, and this is what we call the one door, one key approach. So you submit one dossier that goes to the system, and then the system itself, through EFSA, will divide the work and will take care of the assessment for the environmental assessment to be done under the Directive 2001/18 by the authorities of that directive. The other option is, of course, because this is an option, it's not an obligation, is that those dossiers are submitted separately. They can be separately or consecutively. So first for food safety, for example, and then for cultivation, or the other way around. With the group, the conclusion has been drawn that for this honey suite, it will go in the second way. First of all, is submission for the cultivation dossier through the Dutch competent authorities, following the Dutch rules first, and then it will be submitted to the EU system. And then later on, if we have a bit more time, then there will be a submission of a food feed dossier, most likely through the competent authorities of the Czech Republic, done through the colleagues in the Czech Republic. So just as a heads up, the dossier will go in two steps. First, about environmental safety under the Directive 2001/18, and then for food feed safety under Regulation 1829. Then the question, who will submit, was a long discussion, because there are a lot of institutes involved, and it could be one of those, but it can never be the combination of those. You cannot, as a group of research institutes, submit a submission. You have to be one legal entity. So the conclusion was that for this purpose, we established an international non-profit association. Originally, this was the Honey Sweet Association, then we received more and more requests, whether later on also other specialty crops could be submitted. So this has been expanded to the Public Research Specialty Crops Approval Associations. Difficult name, PRESCA, but this best describes what it does. The purpose is, as with all these associations, the main thing is they collaborate, and they collaborate for the purpose of facilitating the availability of genetically modified crops that are developed by the public research sector. The activities will be primarily preparing and submitting dossiers, and thereby just gaining the experience, but a whole number of other things can be done just under this association. So it will be the association that will submit the dossier legally. And then the last part, a lot of this work is done by volunteers from many directions. A lot of scientists, left and right, have contributed their time. Organizations, companies, we had law firms helping with the association, consulting companies on public research for the outreach and so forth. The list is too long to mention it, but let me just highlight one scientist, and that's Nick. And Ralph, the name is Nick van Geluwe. And Nick is a PhD student who had his PhD last year in December. And during the course of last year, he heard about this dossier, and he offered to help. And this is highly appreciated, because as you've seen, there is decades of information collected in the US, in Europe, and this all has now to be galvanized in one submission, and then still adhering to all kinds of other rules. And Nick will explain that. So Nick, on behalf of the entire association, thank you very much for what you're doing. And with this, I'm happy to hand back to Ralph to pronounce Nick's name one more time.
Okay, well, Nick, I apologize, but I'm not even going to try at this point. But I will repeat that you recently obtained your PhD at Ghent Center for Plant System Biology. You studied mechanisms of lateral root development using genome editing with CRISPR. You gained experience in regulatory aspects related to plant biotechnology, as well as teaching plant biology and science communication and outreach activities. And with that, I'll turn it over to you.
Thank you very much, Ralph. Good afternoon, everyone. It's my pleasure to present to you let's say, the work that has been done and that is going to be done related to the preparation of the dossier for cultivation. So my name is Nick van Heluwe. I recently finished my PhD. So I won that, let's say, battle. And the next challenge that I'm working on in my free time is this honey sweet dossier. And just to give you some background, so I have background in plant biotechnology, but of course, we do many great things in the lab, but I think there's also an important societal aspect. And that's how I got in touch with Piet, and I was trained by Piet into the world of the plant biotechnology regulations, which are still pretty complex to me. But this is, I think, an ideal test case for me, and I think for many fellow researchers and other people who are interested in this, to see how we can try to introduce something to the market. So as Piet indicated, the procedure is that PRESCA, the association, will be the applicant. And in this case, the national competent authority where the dossier will be submitted is the Netherlands. And so currently we are preparing a dossier for cultivation of the honey sweet plum in the EU. So this application form from the Dutch government is available via their website. And if you would download this, you see this is only 6 pages long. However, if you see what is asked of us to provide such a dossier, the pages increase tremendously. And I think I can share that we're over hundreds of pages now with the information. I'm not going to show you that, but I'm going to highlight here you through the different steps of such a GMO dossier. And what is the outline of such an application form? So in the first chapter, we will provide general information. Then next, we will describe the nature of and this abbreviation stands for genetically modified higher plants, GMHP, that is so contained in the product. Then we will provide information relating to previous releases of this GMO and also information relating to a monitoring plan. So let me first start with the first chapter, the general information. So this is just here to give you a taste of how this dossier looks like. So we have to indicate in which country, what is the name, who is the notifier. So this is this association, and also provide additional information. What I found interesting is if you prepare such a dossier, there is a substantial amount of information asked. And for example, is also asked there in the dossier, so where or for example, plum trees currently being grown, and how about the amount of plum that is being produced every year and exported. And what I found very interesting to see is that actually in many European countries there are plums being produced, but the one that really beats every other member state, that is obviously Romania, which produces about half of all the plum across Europe. But then you see it comes also really to rather quite technical things that are being asked. So for example, we indicate in this dossier that honey sweet plum is intended to be used as conventionally produced plum trees. So the rationale, if you would go deeper into how such a dossier of how the GMO directive was designed, is that you compare the GMO with, let's say, the closest possible plant that you compare. And so in this case, it is the non-transgenic plum. So then in a second paragraph here, they ask, so the type of users of honey sweet plum, are they going to be the same? Yes, they are going to be the same as the type of user of conventionally produced plum trees. And what is your intention? Well, honey sweet plum is intended to be cultivated as conventionally produced plum trees. And also, it is intended to be confined under the terms of the authorization applied for all EU countries. And here you see also additional information which is actually relating to the opt-out strategy. I will not go into detail, but here I want to show you have to clearly show your intentions of the GMO product in this application dossier. Then, of course, you have to explain the nature of this genetically modified higher plant. And there are several topics that are here being addressed. And I can share with you that actually this chapter really constitutes most of the dossier. So let me start with first giving information relating to the recipient. So here we are working with honey sweet, and this honey sweet is so the transgenic version of this cultivar, Bluebird. So this is in particular cultivar that was chosen to genetically modify, and this is hexaploid and is also self-sterile. Then in that dossier, you will describe what the possible sexual compatibility is between cultivated species, but also, for example, between wild species. And so let's say, so for this prunus, and all the possible relatives, there are quite a number, let's say, of possibilities. And also even geographical indications are required in that case, which is also very, let's say, interesting to learn about, in this case in Europe. Then you also have to provide information related to the survivability, the dissemination, and the interaction with the environment of this plant. And then also general phenotypic and genotypic traits are being discussed. And for example, there was a question here during our iPlanta webinar about, what about almond, for example? Well, almond is diploid. And so many of other, let's say, cultivated prunus species, they have a different ploidy compared to the plum. And as of a result, so the pollen of this plant can indeed lead to an interaction with another species, but they will not result in an offspring because there is a different ploidy number. So this is relevant information that need to be provided in the dossier. And then this is a bit, let's say, closer to where my background is, but although it's represented in a more complicated way than I'm used to, that is, of course, the information related to genetic modification. So actually, a tDNA construct was designed to make it possible to use agrobacterium for the genetic modification. And so here you see depicted the tDNA with on one side the right border and the typical left border. And here we have also the typical, let's say, genes that are encoded in this factor. And so the genetic code that is responsible here for the function, it is this Plum Pox virus code protein gene. So we describe completely the plasmid, the tDNA, the agrobacterium, and then also how we obtained this genetically modified plum tree. And it was through the co-cultivation of hypocotyl slices. And then from on that, a complete new plum plant was regenerated. So this is information about the genetic modification that was introduced. Of course, then the transformation happens, and that is a kind of a black box because I talk from my experience in the molecular lab. We usually don't care. We are happy if the plant is genetically transformed, for sure, in a case like with Arabidopsis, and there it stops. We're happy, or we're studying the function that we're interested in. So here you can see, and Chris actually highlighted it already, that, and this is very surprisingly, that 2 insertion events happened in this particular genetically modified event, this honey sweet. So the first insertion, well, it contains 5 partial copies of the tDNA construct and 2 complete copies. And then a little bit further in the genome of plum, a second insertion event happens. And here we have two partial copies, which by serendipity resulted in a hairpin structure. And so, and this is actually the construct design that results in a hairpin structure of double-stranded RNA, and which results in the RNA interference pathway. So this is really an, let's say, event that just happened by chance. It has been described that insertion 1 and insertion event 2, they co-segregate together. So they are very close to each other, and it's not possible to segregate one from the other. If CRISPR, for example, would be possible to use to cut out the first insertion, well, I'm of a true believer that with CRISPR you can do many things, but yeah, I don't know practically how feasible this would be. I would need to look, or we would have to look deeper into that. So here you see then further that we look into where are these insertions exactly in the genome. And then I remember also from other GMO dossiers that I let's say, followed or was introduced during courses, that through bioinformatic analysis, we can look if, for example, no native genes of this plum are interrupted or whatsoever, or doesn't affect, yeah, or that the insertion event does not result in any other unintended effects. And so besides bioinformatic analysis, you can do also, of course, even RNA sequencing, etc., to see if the expression of genes is not unaltered. So in the dossier, we provide different experimental approaches to, let's say, pinpoint on that. So besides this Plum Pox virus code protein gene, there were also other genes present on the construct that help to follow up what is happening in this transformant. So we have, for example, the GUS gene, and so of that was measured a moderate level, and different kinds of tissues have been tested during different conditions, different in greenhouses, field trials, etc. And also the NPT2 gene, which constitutes a resistance to kanamycin, shows a pretty low expression level. And then, for example, also some bacterial elements that are typical for a plasmid, there could no expression have been identified. But what is actually the key message here is that so this absence of expression of the code protein from the Plum Pox virus, that has been maintained for over 15 years of field trials. And this is what I find so remarkable about this particular dossier, is the amount of knowledge that has been gained over the years, and where field trials have been so essential to provide this data. So then further to that, okay, we went from DNA level to the messenger RNA level. And then this immunoblot is actually showing that no protein, no code protein of this Plum Pox virus can be identified in the honey sweet GM plant. So this is also indicated as the C5 construct as compared to other plants here as well, and also a control. And here you have, so as a, let's say, positive control, a different amount of concentration of this protein. So it's completely absent. So in summary, the honey sweet plum, it does not express the code protein from the Plum Pox virus at detectable levels. And this eliminates actually the concern of protein exposure to non-target organisms. So when you work on such a dossier, it's really getting used to this kind of, let's say, phrasing and terminology. Then in the next step, there is more information asked related to the genetically modified higher plant. So for example, based now on this function that has been introduced in honey sweet, how does this differ from the recipient plant? So does this have an effect on the rate of reproduction, on the dissemination of this GMO, on the survivability, etc.? And furthermore, or is there a potential for the transfer of genetic material from this GMO to other organisms? So that all has to be in a structured manner being evaluated. And here we come actually down to risk assessment in that sense. So based on the knowledge that has been built, we show that gene transfer to naturalized prunus species in the EU is very limited because of those ploidy differences and also because of the limited success of interspecific hybrids, because like hybrids can also be generated by people, and that has been done using artificial ways, but it's, as described in our dossier, very difficult to obtain that. And furthermore, there is no evidence at the moment of gene flow from honey sweet plum to microorganisms. And then here I show you something that I would never think of really just as a plant biotechnologist, but these are like the things that are expected of you to describe in a risk assessment. So this is the horizontal gene transfer pathway to harm. So this is a pathway. I think it's outlined and you can find it back via EFSA. You can also always reach out to me or the association to have more information on that, where via this pathway is considered, is there a plausibility that DNA from plum will end up, for example, in soil bacteria. So here we define that the likelihood that the cultivation of honey sweet plum in the EU will result in harmful effects in human or environmental or animal health as a consequence of horizontal gene transfer is negligible. And then finally, we also have to describe information on the potential environmental impact when this genetically modified plant is released. So, and here we come to really an extensive risk assessment that is required of us and is stated in this Dutch dossier. So indicate for each of the 9 areas of concern as given in this annex, why there is a concern or not. And here I depict just a few a few areas just to give you an idea. So the first one is, what is the likelihood of this honey sweet becoming more persistent than the recipient in agricultural habitats or more invasive in natural habitats, and their aspects such as hazard and exposure are very important in your risk assessment. And then a second question, for example, is there any selective advantage or disadvantage conferred to this honey sweet? And so each of these 9 areas need to be addressed and need to be hypothesized on if that would be the case or not, and why we would believe that this is the case or not. Then I come after this extensive chapter B to chapter C, and this is information relating to previous releases. And yeah, I'm very happy that we can actually provide their information, because we have information on previous releases coming from the US, where the first field testing, I think, started in 1995. And now this honey sweet plum has been growing in the US since 2007, even under an unregulated cultivation. And so thanks to the team there from the USDA, we have gained a lot of knowledge there on how this honey sweet tree behaves in the environment. And besides that, a lot of field testing has been going on in the EU. I'm really looking forward to see once myself the field trials. And so across different member states, a lot of data has been generated over the years. Nevertheless, all of this actually leads us to conclude, so no immediate or delayed adverse effects as a result of the direct and indirect interaction of the genetically modified plum with the environment have been identified. And then the final chapter of this outline is information relating to a monitoring plan. So a general surveillance plan is mandatory. That would be, for example, in the case that unforeseen adverse effects are caused by your GMO. And so we provide such a plan in those dossiers. And then it's also possible that you need to provide a case-specific monitoring plan that might be also as a result of the risk assessment, where you think that there might be some high potential for certain, let's say, adverse effects, but that is not considered necessary for the honey sweet dossier. So let's say there has been already a lot of work done. I recently finished my PhD, which was a tough job, and now I'm able to dedicate more of my free time to this dossier. I am really impressed by the amount of knowledge and let's say data that has been provided here. Furthermore, we keep on having work because, for example, the EFSA has now or is implementing now new transparency regulation, of which our dossier will also need to fulfill the required criteria. So this regulation is called the Transparency Regulation, and it's one on the transparency and the sustainability of the EU risk assessment in the food chain. So it has different goals. It wants to ensure more transparency. It wants to increase the independence of studies. It wants to strengthen scientific cooperation and develop comprehensive risk communication. And this will start to be effective in March 2021. And so we will have to make sure that our dossier fulfills that, because we plan that this dossier will be submitted in the first quarter of this year. So a link I provide here below with more information. This leads me to thank the association and also the input I received from many members of the association. Also, I want to thank in particular Nancy Podevin for doing this extensive molecular characterization in our dossier. And if there would be any questions or you would have any ideas or feedback on our current work, please do not hesitate to reach out to me, to the association. So I want to make clear here that I am here interested in this project because of the biotechnology and also the regulations, and to provide something for society, and that I am here mainly the processor of all this data, knowledge and text that has been provided over the last years. And with this, I hand back to Ralph.
Thank you very much, Nick. That was another excellent talk. Very well done. We have some questions that we can discuss. Pete, do you have anything to say at this point?
I'm glad that you give me the floor again. Two things, because what is particularly challenging about this dossier is that it has a long history. And this meant, for example, that the forms that Nick is using for from the Dutch government have not been used for many, many years, because ever since this one door, one key procedure was opened, that you could submit all the aspects in one dossier, none of the governments have actually worked on this anymore. So we have been talking with a lot of EU governments. Some of them could not even find the forms anymore that they have been using for so many years. So we're working with very old forms that have to be interpreted and worked at in the light of the new requirements. So that is the first one. The other aspect is that when these plums were sent to Europe for testing, the tests were mainly done for efficacy, for scientific interest, but in regulatory terms we call nice to know. And so the challenge here is to translate all those data that have been actually collected for a different purpose now into regulatory dossiers. So this is it's a very challenging thing, and I understand, or I hope that you will all understand why we're happy to have Nick. And I take this opportunity to invite anyone who would be interested in helping us out, even in small pieces. That'd be great. And as Nick said, even what looks like a small piece in the dossier, the molecular characterization, Nancy has been working on that for days, if not weeks. because we need to present it in a way that is conform the requirements of the EU. So I take this again as an opportunity to invite anyone who is interested in participating, volunteering some time. We're happy to chip off little bits and pieces to others to work on. And with that, I give back to you, Ralph.
Okay, Pete, a question though, if someone does want to get in touch and offer something or have some questions, what would be the best route for them to get in touch with the association in general? And then it could be parsed out to the best person to answer.
At the moment, it's best. Nick had his email address on his last slide. Send it to Nick, and Nick will take care of what comes to me and what goes to others.
Okay, terrific, great, thank you. So a couple of questions or comments, and I think most of them have been answered. For example, could CRISPR be used to remove the insertion event? I think Nick answered that. About almonds being pollinated from plum, they're not compatible, so a seed wouldn't form in that case, except maybe in a very, very, very rare case. So basically you wouldn't have an almond being formed. An almond nut being formed. Is it possible to get rid of the insertion event? Is the NPT2 present in honey sweet? The NPT2 is present. I think we saw that, and it's very low expression, extremely low expression. About getting rid of the insertion event through CRISPR, that was answered again by Nick. Do you think a different gene construct, like a hairpin-based gene construct, could induce different responses? Perhaps sometimes those hairpins work and sometimes they don't. But if anyone else wants to try and answer that one, please do. Several of the data for the EU dossier presumably have been generated only during the past few years. However, honey sweet has already received approval in the US in 2011. Can it be concluded from this as several of the newer data may not be necessary for safety assessment and are just nice to know?
Ralph, this is Pete. I'd like to respond to that with a question probably to Nancy, because if I understand well the transparency rules, the new rules, the whole concept of nice to know no longer exists. Everything that you have has to be submitted. I hope I can't see her, but I hope that Nancy is still on the line because she's quite experienced in this. Nancy, are you still with us?
Yes. I mean, I don't think this is coming from transparency. So under the implementing regulation in the EU, it was already indicated that we as an industry need to submit everything that is relevant. Question is always what is relevant. And so we often provide whole lists where EFSA then picks and chooses which studies they think are relevant. So I don't think transparency is going to make a difference. I think the implementing regulation has made it much more difficult for public institutes to actually get into the game, because they are now asking that all studies should be done GLP or ISO. That is not a trivial thing. I mean, that has come in the GMO legislation, and I think it actually gives a barrier and actually makes that only big companies are able to comply, because also the number of studies that have to be done in the EU are much more than in other geographies. And that's why you're also seeing that in other geographies, like in the US, that you have smaller players actually on the market. While I find it very unfortunate that actually in the EU, we've now seen new companies coming in and submitting dossiers. They are not able to even get through completeness checks. So EFSA first checks, is everything that legislation asks in your dossier in the proper manner? And so I think there have been 3 or 4 applications submitted in 2019 and 2020, and they have been stuck for more than a year in completeness check and even not been able to reach the risk assessment phase. And so that seems to be privileged to big companies that are able to put 15 million into an application, which also makes that if you have a smaller crop, to get 15 million again out of selling that crop is probably very challenging. And that's why I don't understand what EFSA always says. We don't understand, we only have row crops. and these kind of herbicide tolerant and BT events, but it is because that's the only place you're able to get that much money, again, from selling the product, and other trades and crops don't really have that possibility. To get the money, you have to invest in out of it, which I find very unfortunate. I mean, I've always been happy that I've worked with the smallest company in the business. I would like to work for a smaller company, but those don't seem to have a chance in Europe.
Well, thank you, Nancy. That's we're just going to have to see how this all goes, aren't we? But there has to be some. If we're going to improve crops in a world that's undergoing so many catastrophes, climate change, et cetera, the invasive species, we're going to have to use new technologies. We can't. I was a fruit breeder for my entire career, and the time that it takes to, for example, release a new fruit crop is really decades. And in this world now, I'm not sure we can afford decades to solve some of the critical issues we have with our crops, with our forest species, et cetera. So I think there needs to be some kind of change of attitude and realization of of what our needs are in terms of a planet and of an agricultural community. So with that, let's see if there's anything else here. Yes, as honey sweet is approved for cultivation in the U.S., but is actually cultivated, but is it actually cultivated there? There seems to be no need. So no, it's really, it's for sale. It can be purchased in the U.S., but it's not in commercial production. I think there are some homeowners and things like that who are people just interested in genetically engineered crops, growing some trees. But really, its main use now is being as a germplasm resource, and it is being used in breeding programs to develop additional varieties that have the resistance. And the resistance is inherited as a single dominant gene trait. So it's very easy to select for. Also, since it does express the GUS gene, it's extremely easy to pick out the seedlings that have the gene because they're GUS positive. So this GUS gene, I think, is kind of interesting because it not only helps breeders, but it also, in terms of looking at any kind of issues related to issues that EFSA might be interested in, it's easy to see if there's some tree that someone suspects might be genetically engineered, honey sweet, and to see if it's GUS positive. It's not GUS positive. It's not honey sweet. So that's kind of an interesting sidelight on that. If anyone sees a question that they want to answer, please jump in. I'm just going through them and seeing what we have here.
Ralph, I see there another question. All great presentations and impressive initiative to put our full regulatory dossier together. Will the team also develop a communication plan and key messages regarding the event, as this will attract a lot of media or even activist attention? So yeah, we have some members in the association who are really forward-looking, and so we are in contact, and we have actually the involvement of a consultancy company that will help us with that, because we think indeed this is very important when this dossier will be submitted, that the public is informed and involved about this.
Yeah, and Ralph, this is Pete. Let me add to that it's indeed great that we have had help from law firms, and indeed a very big consultancy, a global consultancy that picks these things up way beyond what any public research institute can do. Right? They make outreach plans and media plans, and if you see the whole approach, and they do that just to help. They do this pro bono, which is great. And we're working on a website, and at one point we will have all of that on the website, because we do want to share this. This is the ultimate open science concept, so to say. Whatever this group has gained in experience will be shared. And let me add one more thing to make it perhaps even more interesting for people to join in with some volunteer work. Is one of the things that we need to do, and Nancy referred to this, is that if more data is requested, big companies provide the data. Despite that they cost a lot of money, despite that they will all tell you that scientifically there's no point in asking those data, they will provide it. And that's for understandable reasons, because for them it's comparing getting a big return on their investment or none at all. But the extra tests that are being requested sometimes are more than the entire research budget of the entire institute. So this luxury is only with those big companies, not even with small companies, let alone with the public research sector. So one of the things that we have been discussing with the consortium to do in the dossier is to make use of that article in the law that says that if you believe that certain information is not scientifically relevant, then you can claim that. And we believe as an intellectual exercise, it will be very interesting to work that out. To define this kind of information may be nice to know, but is not need to know in terms of safety. So a lot of work will be focusing on phrasing very scientifically, very correctly as to why certain data would not be necessary.
Yeah. Can I say a few things? Yeah, first of all, thanks all. But I want really to strongly support this last comment from Pete, because it's impressive for me that in the last 20 years, almost, if I understand well, practically no dossier have been submitted, and so that for cultivation. And so it's something that, okay, we agree that we don't want GMO in Europe, but at this point, it's also the fact that we are not doing this work. So this experience is something very, very important, not only for us, but all scientists, for showing that this there is a possibility it can be done. Clearly, at the moment, it's very costly, as Nancy is saying. But as you, Peter, are saying, probably through this experience, we can even demonstrate, and on the experience, on science data and so on, that many of the things that have been requested in reality can be provided, probably not spending such a big amount of money, and probably can be even accepted without or consider data that we can provide enough in order to consider this. Clearly, we have to a very strong battle again in front, because we know what is the public opinion, what are the big oppositions that we can expect. But I'm not scared about this, and we should face this opposition even with this small experience. So I fully support this idea. And then I have also a technical question that comes through about the construct and the harping. It related in particular, and that for me is another important issue specifically related to gene silencing technology, the possibility to use the rootstock. So from the preliminary data that you have, in some way, this you are saying that apparently for RNA suite is not working. But in reality, you have a construct that in some way has been at the beginning prepared with a different approach. Then it turned out that it's a double-stranded RNA, a harping sequence. But yeah, again, that's my question. We should really focus on the fact, trying to understand if the new construct, if well prepared, well defined with some specific approach, can really improve the capacity of having gene silences through rootstock in fruit tree. That is very critical, not only for plum, for peach, but for practically, I would say, all the fruit crops, because we have many problems with fruit crops that can be controlled in this system, not only virus, but also pest and diseases that can be controlled. And in my opinion, that remains really a critical and fundamental point that we need to demonstrate. There are some indication I know on cherries as they are working on that, they are providing some results of that, but we need to really focus on this work, and any information available are really welcome for that. Thank you.
Thank you, Bruno. I'm looking at some of these other questions. There was a question about only 2 insertion events were found, considering the entire genome. and it says agrobacterium transformation is known to possibly generate more insertion events throughout the genome. I think, Chris, you might want to address that. I think you really already have in your talk.
Yeah, I can comment. So we had over 300x coverage from Illumina sequence, and we have another roughly 30x coverage from nanopore data. We have searched the entire plasmid that was used for potential other insertion events, even outside the tDNA borders. We didn't find anything. We even searched the entire agrogenome to see if there was anything. We didn't find anything. So based on all the data we have, we're very confident there are only 2.
Great, thank you.
I have just 3 additive comments, Ralph.
Excuse me?
May I have to give some 3 additive comments?
Sure.
Yeah, okay. There are 3 points that I want to, precise. First of all, about the prunus crossing. Nick titled in his talk about the limiting factor about the prunus crossing. There is one key point that has been forgotten, is the asynchrony. The C5 is blooming at the different time to the majority of European prunus here. This is one point. The second is about the interspecific cross. The interspecific cross can be done, but only through artificial pollination. You force, otherwise it cannot work. And this is really experimental. And the third one, in the comments about why C5 doesn't express CP, even if there is one insert that is untied. The reason is simple, is the RNA dependent polymerase 6 also is degrading certainly the RNA that is transcribed by this insert. And that's the reason why you don't have the protein. So the plants have this device very powerful to clean up cells about the protein expression, similarly like also the PPVCP expressed when the plant is infected. It's similar like that. That's the 3 points that I pointed.
And perhaps the expression of the coprotein by that one insert actually keeps the process always turned on. So the plant is always getting something that turns on the silencing system. I wonder about that.
The silencing is very powerful.
Yeah.
RNA is more.
Oh, go ahead. Let's say.
There's another question I see there, Ralph. Like, will the presence of the NPT2 and the GUS gene cause problems in the regulatory process? I will try to answer based on what I think. I think there are already similar, let's say, dossiers that have been submitted with this NPT2 or this GUS gene. It can be that it's already described that this will not have any, let's say, negative or adverse effects. But I'm looking actually again at Nancy for this, what she thinks based on her experience.
Yes, NPT2 has been in different events. I think the last one was the Monsanto event, also including a cold shock protein. That application had a real hard time, criticized a lot by the NGOs. Also the legislation in Europe has said that superfluous DNA should be avoided. So anything which is not necessary for your trait should not be included. So in principle, you would say the main problem with NPT2 is horizontal gene transfer. And as long as you do not have the possibility for double homologous recombination due to flanking sequences to bacteria, this risk has always been considered very small. You will always have a case-by-case assessment to look if double homologous recombination would be possible. But if the conclusion would be that it's not possible, from a risk point of view, you would conclude it's not negligible. From a public perception point of view, I have seen that this is challenging.
And Ralph, if I may, this is Pete. No, I think Nancy is right. There is the distinction between the public perception and the technical risk aspect. These are almost topics for entire PhDs, because the whole question of what is superfluous DNA is, I think, is a fantastic one. And the other one that is being raised very often is that there's a policy in Europe that antibiotic resistance genes that could pose a risk for medical treatment should be phased out. But that as such, because NPTT is not part of that, EFSA has looked at it multiple times. And this is again why this might be an interesting dossier to see whether EFSA's view has changed or whether EFSA is willing to come to a different conclusion on the same data and so forth. So this will be very interesting to see whether the whole question of horizontal gene transfer and the already very high presence of kanamycin resistance of NPT2 genes in the environment, whether that will be looked at very differently. So again, from a scientific perspective, this will be a very interesting case to take it forward. And this whole notion of superfluous DNA, I love it. I'd say if I were young, I would go and do a PhD on that particular topic.
Well, I think another topic which actually would be very interesting, in my opinion, is that our legislation is still based on the precautionary principle. That principle came in the 80s because we had limited information regarding what it is when we do transformation, what it does. Actually, we have now a lot of information. I mean, we have shown that just inserting DNA is a natural process, and that's also done in nature. So that's not really something very specific. But we still have this precautionary principle in our legislation. And in principle, precautionary principle is only used in those areas where you have a lack of information that would make that certain hazards would be overlooked because you don't know anything. We already have 20 years now of molecular biology on our side to actually say we should move this from a precautionary-based approach to a communication approach, because the main challenge we have in this domain is that if you read an EFSA dossier and you read the opinion, there is no unknowns highlighted. So the precautionary principle is not applicable because nobody is saying that there's still something to be analyzed and we need more data to actually be able to conclude. So now the main problem we have is the difference between the actual risk, which is coming from the risk assessment by the scientists, which are saying it's as safe as, and the public that actually perceives a risk and perceives it's risky. And so for me, this makes that actually risk managers should move from a precautionary-based approach to a communication-based approach, because you will not resolve this issue by always asking more data, because people don't know how much data companies are actually providing to EFSA. It doesn't make it safer, it just makes that we have to slaughter more animals, which is also in principle something that European citizens also don't really want. So at a certain moment, we need to convince our risk managers to just start communicating that this is not really a hazardous thing to do, and that the perceived risk is not, well, correct, and that people should adjust their way of thinking on this technology. And I think that we really need this, because if Commission also don't start to communicate on, for example, gene editing, we might risk in the EU to end up in the same situation we end up with GMOs. And we have seen, like in countries like Belgium, where your government started to communicate more positively on GMO, that this has an impact on how people are viewing it, that they're starting to see the benefits. But if our risk managers keep on not being present in the debate, I don't think we will ever resolve this, because if we as big companies communicate, there are not that many people that are willing to believe us.
Bruno, I think that Nancy just outlined the next 5 webinars for iPlanta. So,
yeah, no, this is part of our program. But I think, and again, I want to thank Ralph, Pete, and everybody from the Honest Sweet group, that this is an example. This is a case study, this is an example. We need to bring out many other examples, because I think that is, again, wasting of time, keep going around, say, look at this, discussing on that, and so on. But we can really probably change a little bit the things if first, I keep saying this concept, first we show the product, then on the product, with the growers, maybe with the consumer, we start to speak with the policymakers. Because I know that growers have a lot of problems, and we need to show that this technology is solving that problem. And if we get the support of at least, at the moment, if we go out, we have, at least in Italy, but I think also in many countries, many of the grower associations say, no, we don't want, we don't support you, or it's difficult to support you. If we can show that a few examples that can be very helpful for solving problems for the growers, and then after keep using this in your communication, I think that it will be much more impactful. And clearly, again, we should try really to work with a small company. We say small company, but you know, at least in the fruit sector, are a small company, but they are not so small. Clearly, are not so big like a large multinational. But if they can start to support, they are starting to support projects, at least for us, it's like that. Clearly, they are not giving us huge amount of money. They want to see something. If they see something, if we can demonstrate that, for example, and I know there are people following here now today, the fact that we can solve the Sharka problem in peach in Italy, we can have a really strong support from Italian association, Italian company, and probably also public institution, because there are regions that they are facing strong problems related to the Sharka disease for peach cultivation. That it was one of the main crop, and now they are losing totally almost. They are changing and moving to other, they have to change and move into other crop because they don't have the possibility to maintain peach. So again, let's keep going. I think that there are possibilities. Probably we need to change a little bit our approach. And yes, keep following with, I will keep you sending emails reminding the next webinars. I don't know, if you want to hand Ralph, then I want to say a few things about the webinars. And then after, if all of you switch on your camera, I can take a final picture.
Yeah, I think we've answered all the questions, or if not most of the questions. So I think we can go ahead and give the floor to you again.
Yeah, well, thank you. I want also to thank Ralph again, and all the speakers for this very nice initiative. And yes, as I was saying, I will keep you informed about next seminars. The next one, it will be on, again, on specific topics, like we had today, the virus. Next, it will be fungi. 11 of February, and then insect. We have a new program still not available on the web page, but it's ready. Last night we were asking, looking at the last confirmation for the speakers. And then there will be two important webinars, one on communication that Hilde, you and Jeremy are preparing, where also Mark Linas, you know Mark Linas? Mark Linas is a writer, he was from Greenpeace, and he wrote a book in support of GMO coming out from Greenpeace. So in some way, it would be interesting to have his opinion in terms of on communication. It will be very rich webinars with many communicators. So I think that will be very interesting to know about that. And then another one, we are working on it. It's on the stakeholders. We are trying to involve as much as possible scientific officers from the European Commission, but also stakeholders, so representative of small groups, a small organization, and so on. So also from your side, if you have suggestions, we are still working on the program. So you are really welcome to approach us and suggest, I don't know, to involve the president of a bee association, or of a peach or plum or potato association and somewhere. I think that it will be very, very helpful to have these people involved, so to bring them this discussion. Then the epigenetics, that I think that's also another important topic. And then we will be at the end of iPlanta. So thank you again. If you turn on your camera, and also you go in front of your camera, we can have the full screen. Yes, now I see a full screen, so I can push and have a picture. And so thank you very much. And yes, this is finished, and we will be in touch. Again, the material has been recorded, and it will be available on the web page. So in case some of you want to listen again, part of the work done today, you are welcome. Have a nice evening, and see you next soon on the web. Bye-bye.
Bye-bye. Ciao, ciao.